Good evening, ladies and gentlemen. So today we're learning about microscopy. So this is part of our unit on general microbiology. So I may ask someone, okay, before I ask a question, you can look at the main objectives. So we are expected to define what a microscope is. You should also be able to identify different parts of a microscope, explain their functions of each of these parts, and also you should be able to describe the various types of microscopes. So I will start by asking someone, what is microscopy? You unmute yourself and speak. Okay, so it is just the technical field where you are using a microscope to view images. Organisms can be microorganisms. So, and the main function is to magnify them to make them appear bigger so that you can see each of the smallest part of that microorganism. And we say the small organism that we cannot be able to see using our naked eyes. So that is why you have to use a microscope. And this field is what we refer to as a microscopy. So here is a microscope. I believe from the biology, we all know that there are various parts of a microscope. There is the head, you can clearly see it. We have the objective lenses. There are different types of objective lenses having different magnification powers. We also have like the stage where you place your specimen. You can see we have the adjustment knob, where we have the fine adjustment knob, we have the coarse adjustment knob, and all other parts that you can clearly see from this sketch. So in the next thing, we'll be able to identify or to describe actually the functions of each part that we've seen. So for example, you see the arm. It connects the tube to the base. We can see like here is the base from the diagram. We also have a different part. We have the tube. So it's the main part that contains the lenses, the mirrors. We also have the different objective lenses. So the main function, we say the objective lens is to magnify the image, to make it appear larger. So from our illustration, we have objective lenses. We can see 3 of them. So this, it's where you adjust each of the objective lens having different magnification powers, and therefore you will see the images in different sizes. So the higher the power of the objective lens, of course, the bigger the image. And then we have other parts of a microscope. We have the illuminator, which is the main source of light. We have the stage, it's where you place your specimen to view it. So before, you cannot just place it directly on the stage. You have to use a glass slide, and then you fix the glass slide on the stage, and therefore the light source from the illuminator through the condenser, it reaches the stage, and you're able to observe your image. We also have a condenser which concentrates the light to the stage, and these lights, we say it's from the illuminator. We have also the adjustment knob, the coarse adjustment. You're focusing the image, you're moving it up closer to you move it closer, that is the stage, you move it closer to the objective lens using the adjustment knob. And that is all on the parts and the functions of a microscope. So we also have the different types of microscope, a light microscope and an electron microscope. So the difference is just from the word says the light microscope, it uses a beam of light as the main source of course, the light has to reach the specimen. And the electron microscope, this one you see is a beam of electrons rather than the light as compared to the light microscope. So under the light microscope, where you're using light as the main source of light, of course, we have the different types. The first one, there is the bright field light microscope. We also have a phase contrast light microscope, dark field, fluorescence, and the differential interference contrast microscopes. Under the electron microscope, there are two of them, the transmission electron microscope. You also have a scanning electron microscope. So remember, there are only two, but for the light microscope, you can see there are 5 of them. So before the lesson ends, you should be able to identify all these types of light microscope. You should not confuse between the two. So as we end the lesson, I want to give you an assignment where you should be able to describe the 4 types of objective lenses. So we know there are different types of objective lenses, and there are 4 of them. So you should be able to describe the differences between each of these objective lenses. So any question for me before I end the class? Gladys, do you have a question for me?
Yes, I have a question. When do we use a compound microscope?
They will discuss on the light and the electron microscope. Yes?
Yes.
So I said you should be able to know the difference between the light and the electron microscope at your level. Okay?
Okay.
Mm-hmm.
That is okay.
Okay.
I have a question.
Mm-hmm.
How many objective lens do we have?
There are 4 objective lenses, the oil immersion. So that is the assignment that I'm giving you for today. So I expect you to do the assignment and give it to me.
Teacher, I'm having a question.
Mm-hmm. Yes,
Why
please.
is light microscope called light microscope?
Because it uses light, unlike the electron microscope that uses a beam of electrons. So these you're using normal light. The other one you're using a beam of electrons for you to be able to see the image. Okay? So we end our class at that point. So the assignment, you should submit it during the next class, and you should print it. So a minimum of 2 pages. So with that, thank you for your time. Bye-bye.
Bye.